Welcome back friends. Welcome back again to one more of the lecture session from this channel. So in this lecture session we will be studying regarding competent host and transformation of the recombinant DNA into the host organism in detail. So here, as we all know, DNA is a charged molecule, but whereas the specific cell membrane or plasma membrane which do exist in case of the various living organisms, it is highly selectively permeable. That means it won't allow all the type of molecules to come across it. It allows only certain selected molecules to come across it. And most commonly the specific cell membrane or plasma membrane, it allows only hydrophobic molecules to come across it. But whereas the specific DNA, it is a charged molecule. So here, in order to regulate the successful transformation of the DNA across the cell membrane, there is a requirement of various transformation strategies. So right now we will be studying regarding the various transformation strategies through which the successful transformation of the DNA across the cell membrane in case of the various living organisms which take place. So here, as we all know, transformation, it is the mechanism or it is the specific process through which the successful transformation of the nucleic acid from the external source into the host organism, it takes place. And here basically transformation mechanism, it can be successfully classified into two different types. And these may include natural transformation and the other one is artificial transformation. Is it clear? So let me write it on the board. Transformation is the mechanism or process through which the successful transformation of nucleic acid from the external source into the host organism takes place. Host organism takes place. And here the specific mechanism and the specific process, here it is termed as transformation. And here basically the mechanism or the process of transformation, it can be successfully classified into two different types. And these may include natural transformation and the other one is artificial transformation. Is it clear? So let me write it on the board. Transformation, it can be successfully classified into two different types. And these may include natural transformation and the other one is artificial transformation. So at first, if we consider natural transformation, so here what is natural transformation? So here natural transformation, as the name itself suggests, it is a natural process. So here most commonly natural transformation, it is observed when the specific E. coli cell or when the specific bacterial cell, when it acquires maturity. So here when the specific bacterial cell, when it acquires maturity, under such condition, the specific bacterial cells, they are capable to regulate the expression of specific genes termed as competent genes. So here once when the specific competent genes, once when they are successfully expressed under such condition, here it leads to the expression of specific proteins termed as competent proteins. So here once when the specific competent proteins, once when they are successfully expressed under such condition, the specific bacterial cell, it is ready to intake the specific nucleic acid from the external source into the host organism. Is it clear? So let me write it here. Natural transformation Natural transformation is a natural process. Is a natural process. Natural process through which transformation of DNA from the external source into the host organism takes place. Host organism takes place. Here, natural transformation, it is a natural process. That means when the specific E. coli cell, when it acquires maturity, only under such condition, here the successful transformation of the nucleic acid across the host organism, it takes place. But under abnormal condition, here by taking the advantage of natural transformation, here we cannot transfer the specific desirable DNA into the host organism. So here, in order to understand this particular concept, here we can take the advantage of one common example. So here in case of males, we observe mouse tag only during the teenage but not at the infant stage. So here, why the specific mouse tag it is observed only during the teenage here? The specific genes which are responsible for regulating the appearance of mouse tag, they are expressed only during the teenage but not at the infant stage. So in the same way, if we apply the same concept by taking the advantage of natural transformation, here the successful transformation of the nucleic acid across the host organism, it takes place only when the specific E. coli cell, when it acquires maturity. But under abnormal condition, here we cannot transfer the nucleic acid into the host organism. So here, in order to regulate the successful transformation of the nucleic acid into the host organism on our own desire, here we can take the advantage of one more other alternative strategy, which is nothing but artificial transformation or induced transformation. Is it clear? So let me write it on the board. Artificial transformation. Artificial transformation is the mechanism through which transfer of nucleic acid into the host organism takes place by taking the advantage of various strategies, right? So right now we will be studying regarding the various strategies through which the artificial transformation or induced transformation it takes place. So here, one of the specific strategy through which the successful transformation of the nucleic acid into the host organism which takes place by the means of artificial transformation, it may include usage of a specific divalent cation like calcium. So here, by taking the advantage of a specific divalent cation like calcium, here the successful transformation of the DNA into the host organism, it takes place. So here, how the specific calcium ion, it plays an important role for the successful transformation of the DNA into the host organism. Here, as we all know, the specific divalent cation like calcium, it has positive charge. But whereas the specific DNA, it has negative charge. So here, because of the dissimilarity in case of the charges in between the divalent cation like calcium and as well as the specific DNA, here the successful transformation of the DNA into the host organism, it takes place so efficiently. Is it clear? So let me write it here. one of the specific strategy through which the transformation of DNA into the host organism takes place through artificial transformation may include the usage of divalent cation like calcium. Divalent cation like calcium. Is it clear? And the other specific strategy through which artificial transformation it takes place, here it is nothing but subjecting the specific bacterial cell to the heat shock treatment followed by cold treatment. Is it clear? So let me erase the things here. Here, as already mentioned, the other alternative strategy through which artificial transformation it takes place, here it is nothing but subjecting the specific bacterial cell to the heat shock treatment followed by cold treatment. Is it clear? So let me write it on the board. Artificial transformation most commonly it is observed by subjecting the bacterial cell to the heat shock treatment at 42 degrees Celsius. Followed by cold treatment. So here initially the specific bacterial cell it is subjected to the heat shock treatment at 42 degrees Celsius, and instantly the specific bacterial cell it is subjected to the cold treatment. So here because of the variation in case of the temperature, instantly the specific bacterial cell wall it becomes so fragile. So here once when the specific bacterial cell wall it becomes so fragile, now here the specific nucleic acid it is capable to enter into the host organism so efficiently. Is it clear? And the other alternative strategy through which artificial transformation it takes place, here it is nothing but micro injection. Is it clear? Micro injection. So let me write it here. Micro injection basically takes the advantage of fine needle-like structure to transfer the DNA into the nucleus of an animal cell. So here by taking the advantage of a specific fine needle-like structure, here the successful transformation of the DNA into the nucleus of an animal cell takes place. And here micro-injection, it is the specific desirable technique which is most commonly used in order to regulate in vitro fertilization. So here in order to regulate in vitro fertilization, micro-injection, it is the most commonly used desirable technique. And the other alternative strategy through which artificial transformation it takes place, here it is nothing but particle bombardment or biolistics, or in other terms here we can consider it as gene gun. Is it clear? So here by taking the advantage of particle bombardment method or biolistics method or gene gun method, here we can transfer the specific DNA into the host organism. So here basically the specific particle bombardment method, it takes the advantage of a specific device termed as gene gun. So here within the specific gene gun, specific microparticles like gold and tungsten, they are successfully coated with the foreign DNA. So here once when the specific microparticles like gold and tungsten which are coated with DNA, once when they travel at higher velocity and once when they bombard the specific biological cell, under such condition the specific DNA which is coated on the surface of the specific microparticles like gold and tungsten, they are successfully transferred into the nucleus of the biological cell. And here it is termed as particle bombardment method or biologistic method or gene gun method. Is it clear? So let me write it here. Artificial transformation is also regulated by taking the advantage of particle bombardment or biolistics method or gene gun method. So here in case of the particle bombardment method, by taking the advantage of a specific device termed as gene gun, here the specific microparticles like gold and tungsten which are coated with the specific DNA, they are successfully transferred at higher velocity into the nucleus of a biological cell. Is it clear? So let me write it here. In particle bombardment method, by taking the advantage of gene gun, by taking the advantage of specific device like gene gun, microparticles like gold and tungsten which are coated with DNA, travel at higher velocity into the nucleus of the biological cell. And here it is termed as particle bombardment method or biolistics method or gene gun method. So right now we have studied regarding the various tools of the recombinant DNA technology, and right now we will be studying regarding the various strategies through which the specific recombinant DNA technology, it is successfully organized in detail. Is it clear? So let me arrange the things here. As already mentioned, then after we will be studying regarding the various strategies through which the recombinant DNA technology which is successfully organized in case of the various living organisms in detail. So here, as we all know, recombinant DNA technology, it can be successfully organized by isolating the specific genetic material from the external source. So here, after isolating the specific genetic material from the external source, now here the specific genetic material like DNA, it is successfully subjected to the restriction enzyme digestion by taking the advantage of specific enzymes. So here, after subjecting the specific DNA to the restriction enzyme digestion by taking the advantage of specific molecular scissors like restriction enzymes, now here we can obtain multiple DNA fragments. So here, after obtaining the multiple DNA fragments, now here we need to isolate the specific desirable DNA fragment. So here, after isolating the specific desirable DNA fragment, now here the specific desirable DNA fragment, it has to be transferred into the vector DNA. So here, once when the specific desirable DNA fragment, once when it is successfully transferred into the vector DNA, now it leads to the formation of recombinant DNA. So here, after the successful formation of recombinant DNA, now the specific recombinant DNA, it has to be successfully transferred into the host organism in order to regulate the gene cloning or gene transfer mechanism. Is it clear? So let me write it on the board. The various strategies of recombinant DNA technology. Recombinant DNA technology may include isolation of desirable DNA from the external source. And then after subjecting the specific DNA to the restriction enzyme digestion by taking the advantage of specific enzymes like molecular scissors. Is it clear? Subjecting the specific DNA to the restriction enzyme digestion. So here, once when the specific DNA, once when it is successfully subjected to the restriction enzyme digestion, now here we can obtain multiple DNA fragments. Now here, the specific desirable DNA fragment, it has to be successfully transferred into the vector DNA. Is it clear? Transfer of desirable DNA fragment into the vector DNA. In order to form recombinant DNA. And then after the specific recombinant DNA, it has to be successfully transferred into the host organism in order to regulate the gene cloning mechanism or the gene transfer mechanism. Is it clear? Transfer of recombinant DNA into the host organism in order to regulate the gene cloning or gene transfer. So at first we will be studying regarding the isolation of the desirable DNA from the external source. So here how the specific isolation of that desirable DNA from the external source it takes place. Is it clear? So let me arrange the things here. As already mentioned, at first we will be studying regarding isolation of the genetic material from the external source in detail. So here, as we all know, in case of all the living organisms, the specific genetic material here it is nothing but nucleic acid. And in case of the majority of the living organisms, the specific genetic material here it is nothing but deoxyribose nucleic acid. So here, in order to extract the specific deoxyribose nucleic acid from the specific external source, here we need to take the advantage of various extraction procedures. So here basically the specific extraction procedures, it may take the advantage of various degradative enzymes. And here these specific degradative enzymes, they are responsible for cleaving the specific cell wall in case of the various living organisms like bacteria, plants, and even in case of fungus. So here, in order to cleave the specific cell wall in case of bacteria, here there is a requirement of a specific degradative enzyme like lysozyme. In the same way, in order to cleave the specific cell wall in case of plants, here there is a requirement of a specific degradative enzyme like cellulase. In the same way, in order to cleave the specific cell wall in case of fungus, here there is a requirement of a specific degradative enzyme like chitinase. Is it clear? So let me write it on the board. Isolation of genetic material from the external source. Right? So at first, in order to isolate the specific DNA or the specific deoxyribose nucleic acid, there is a requirement of various extraction procedures. And these extraction procedures, they may take the advantage of various degradative enzymes. Is it clear? Extraction procedures may take the advantage of various degradative enzymes like lysozyme in case of bacteria. So here, in order to cleave the bacterial cell wall, here there is a requirement of a specific degradative enzyme like lysozyme. Is it clear? Bacteria. In the same way, in order to cleave the specific cell wall in case of plants, here there is a requirement of a specific degradative enzyme like cellulase. Is it clear? Cellulase in case of plants. And in the same way, in order to cleave the specific cell wall in case of fungus, here there is a requirement of a specific enzyme termed as chitinase. And here, as we all know that the specific genes, they may remain on the surface of a long chain which is termed as DNA. So here, the specific DNA itself, it won't remain within the nucleus of a particular biological cell. The specific DNA, it wraps around a specific histone octamer. So here, in order to separate the specific DNA from the histone octamer, at first here we need to take the advantage of a specific degraded to enzyme termed as protease. So here the specific protease, it is responsible for cleaving the specific histone octamer. So here the specific histone octamer, which is composed of basic amino acids or positively charged amino acids like lysine and arginine. So here these specific histone proteins, they are dissociated from the particular DNA by taking the advantage of a specific degraded to enzyme like protease. So here protease, it is the enzyme which is responsible for the successful dissociation of the DNA from the histone octamer. Is it clear? In order to separate the DNA from the histone octamer, which is composed of positively charged amino acids. Positively charged amino acids. There is a requirement of degraded to enzyme, like protease. Then in the same way, in order to cleave or here in order to degrade the specific RNA which remains within the nucleus of one particular biological cell, here there is a requirement of an specific degraded to enzyme like RNase. Is it clear? RNase is the degraded to enzyme to degrade the nature of RNA. And here by taking the advantage of other degraded to enzymes, here we can degrade all the specific components which are present within the nucleus of one particular biological cell. And then after, if we add the specific chilled ethanol, so here easily we can isolate the specific DNA which is present within the nucleus of the biological cell. So here by taking the advantage of various degraded to enzymes, here we can degrade the various components which are present within the nucleus of one particular biological cell. And if we add the specific chilled ethanol into the nucleus of one particular biological cell, so here easily we can extract the specific DNA which is present within the nucleus of one particular biological cell. Is it clear? The other components are also degraded by taking the advantage of degraded to enzymes subjected with chilled ethanol in order to separate the DNA from the nucleus of the biological cell. of the biological cell. Is it clear? And then after we will be studying regarding how the specific DNA it is successfully subjected to the restriction enzyme digestion by taking the advantage of specific enzymes like restriction enzymes. Is it clear? So let me erase the things here. Then after we will be studying regarding cutting of DNA at specific locations by taking the advantage of restriction enzymes. So here after the successful isolation of the DNA from the external source, now here the specific DNA it is successfully subjected to the restriction enzyme digestion by taking the advantage of specific enzymes like restriction enzymes. So here these specific restriction enzymes they are responsible for cleaving the specific DNA into multiple DNA fragments. Is it clear? So let me write it on the board. Cutting of DNA. Cutting of DNA at specific locations. Specific locations by restriction enzymes. By restriction enzymes. So here, after the successful isolation of the DNA, after the successful isolation of DNA from the external source, the specific DNA is subjected to the restriction enzyme digestion. In order to yield multiple DNA fragments. Now here, these specific multiple DNA fragments, they are transferred into the electrophoretic chamber. And here the specific electrophoresis which is responsible for differentiating or for separating the DNA fragments, here it is nothing but what? Agarose gel electrophoresis. So here by taking the advantage of agarose gel electrophoresis, easily we can separate the multiple DNA fragments. Is it clear? Agarose gel electrophoresis. Agarose gel electrophoresis is responsible for separating the multiple DNA fragments. So here within the agarose gel electrophoretic chamber there is an available of specific mechanism termed as sieving effect. So here the specific sieving effect it is responsible for separating or for differentiating the larger DNA fragments from the smaller DNA fragments. Is it clear? Sieving effect within the electrophoretic chamber is responsible for separating the larger DNA fragments from the smaller DNA fragments. From the smaller DNA fragments. From the smaller DNA fragments. And here, as we all know, DNA is negatively charged. The specific DNA, it successfully migrates towards the anode. Is it clear? As we all know, the specific DNA, it is negatively charged. Within the electrophoretic chamber, the specific DNA, it successfully migrates towards the anode. As DNA is negatively charged, it successfully migrates towards the anode. And here the specific restriction enzyme, which is most commonly used in order to cleave the specific external DNA source, that particular restriction enzyme or that concerned restriction enzyme, even it is also used to cleave the vector DNA. Is it clear? The specific restriction enzyme, which is most commonly used in order to cleave the external DNA source, that concerned restriction enzyme, even it is also used in order to cleave the vector DNA. So here, once when the specific vector DNA, once when it is successfully cleaved by taking the advantage of restriction enzyme, now here we can insert the specific DNA fragment. Is it clear? The specific DNA fragment which has successfully obtained during the restriction enzyme digestion, that particular restriction fragment or the particular multiple DNA fragment, it can be successfully inserted into the vector DNA in order to form the recombinant DNA. So here, once when we insert the restriction fragment or once when we insert the specific DNA fragment which has successfully obtained during the restriction enzyme digestion into the vector DNA, now here it leads to the formation of recombinant DNA. Is it clear? So let me write it here. The specific restriction enzyme, which is most commonly used, to cleave the external DNA source, even the concerned restriction enzyme, is used to cleave the vector DNA. So here, once when the specific vector DNA, once when it successfully cleaves by taking the advantage of the concerned restriction enzyme, now here we can insert the specific restriction fragment or the specific DNA fragment into the vector DNA. So here the successful insertion of the restriction fragment or the DNA fragment into the vector DNA, it leads to the formation of recombinant DNA. Is it clear? So here, once when the specific recombinant DNA, once when it has successfully formed, now here we can transfer the specific recombinant DNA into the host organism in order to regulate the gene cloning mechanism or the gene transfer mechanism. Is it clear? And then after we will be studying regarding gene amplification through PCR cycle. So let me arrange the things here. If you look at the gene amplification through the PCR cycle, so here PCR stands for polymerase chain reaction. So here, by taking the advantage of polymerase chain reaction, here we can amplify or here we can manipulate the concerned gene into multiple times. So here basically the polymerase chain reaction cyclic process, it takes the advantage of a short oligonucleotide sequence termed as primer. And here the specific primer, it successfully recruits to the 3' end of the template DNA. So here in order to manipulate or here in order to amplify the consent gene, initially the short oligonucleotide sequence termed as primer, it successfully recruits to the 3' end of the template DNA. Is it clear? So here the successful recruitment of the primer to the 3' end of the template DNA, it successfully exposes the free 3' hydroxyl group. Is it clear? So let me write it here. Polymerase chain reaction takes the advantage of short oligonucleotide sequence like primer, right? So here the specific primer, which is a short oligonucleotide sequence, it successfully recruits to the 3' end of the template DNA. So here once when the specific primer, once when it successfully recruits to the 3' end of the template DNA, now here successful recruitment of the primer to the 3' end of the template DNA, it lead to the exposure of free 3' hydroxyl group. So here once when the free 3' hydroxyl group, once when it successfully exposes here, now here a specific enzyme termed as Taq polymerase, it comes into the picture. So here the specific Taq polymerase, by taking the advantage of a short oligonucleotide sequence termed as primer, and even by taking the advantage of the information which is present in case of the template DNA, the specific Taq polymerase enzyme, it is capable to incorporate the DNTPs. Is it clear? So let me write it here. So here by taking the advantage of primer, Taq polymerase enzyme is capable to incorporate the DNTPs according to the information in template DNA. Right? So here the uniqueness of the specific Taq polymerase enzyme, here it is nothing but the normal DNA polymerase enzyme, which is most commonly used during the replication mechanism. It is capable to work so efficiently only under optimal conditions. But here under abnormal conditions or under extreme temperatures, the specific DNA polymerase enzyme, which is most commonly used in order to regulate the replication mechanism, it undergoes denaturation. So the reason why instead of taking the advantage of the specific DNA polymerase enzyme, here we can take the advantage of a specific enzyme termed as Taq polymerase. So here the uniqueness of the Taq polymerase enzyme, here it is nothing but the specific Taq polymerase enzyme, it is capable to work so efficiently even at extreme temperature. So that is the uniqueness of the Taq polymerase enzyme. And here, why the specific enzyme, here it is termed as Taq polymerase. Here, the specific enzyme, it is isolated from a specific bacteria termed as Thermus aquaticus. Because of the isolation of the specific polymerase enzyme from an extremophile termed as Thermus aquaticus, here the consent enzyme, here it is termed as Taq polymerase. So here the uniqueness of the Taq polymerase enzyme is the specific enzyme, it is capable to work so efficiently even under extreme temperature. Is it clear? So let me write it here. Taq polymerase is capable to work so efficiently under extreme temperature around 90 degrees Celsius. So here, if we manipulate or if we amplify the specific gene for billion times, here we can obtain billion copies. Is it clear? If we manipulate or if we amplify the specific gene for billion times, here we can obtain multiple, or here we can obtain billion copies of the concerned gene. So here, by taking the advantage of the specific PCR cycle, here we can manipulate the concerned gene for many times. And here, if the specific gene which has manipulated by taking the advantage of PCR cycle, if it is, so here the specific fragment, it can be successfully cloned by taking the advantage of a specific mechanism or a specific process termed as gene cloning or gene transfer. So here this is regarding competent host in detail, and in the next upcoming lecture session we will be studying regarding introduction of the recombinant DNA into the host organism and even the obtaining of foreign product in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.